Good afternoon, grade 9. Welcome back to our week 3 discussion. So the past 2 weeks we had discussed about the respiratory system and the circulatory system. So for today's video, we are going to talk about their heredity, inheritance, and variation. But before we proceed to our discussion, we will first discuss or tackle about the objectives of today's topic. So, you are expected to be able to do or achieve the following at the end of our lesson. So, first we have here, discuss the terms related to heredity and inheritance. Next, distinguish Mendelian and non-Mendelian genetics. Then after that, explain the genetic disorder. And then fourth, discuss the Punnett square. And lastly, practice the Punnett square. So for this slide, we have here a family picture. So the family picture shows how some traits of the parents blend into the traits of the offspring or the children. So can you name some of the traits? So as you can observe, the mother's hair color is the same as her daughter's hair color, which is brown. Then the father's hair color is the same as his son's hair color, which is black. So people have been fascinated at how children will resemble their parents and vice versa. As years went by, scientists began to search for more information on how these traits are passed on. The passing of traits from parents to offspring is called the heredity, and the science that deals with the study of heredity is called genetics. So now let's discuss the heredity. So when we say heredity, heredity is the passing of genetic traits from parent to offspring. Now for this slide, let's talk about Gregor Mendel. So the basic laws of heredity were first formed during the mid-1880s by an Austrian botanist monk named Gregor Mendel. Because his work laid the foundation to the study of heredity, Mendel is referred to as the father of genetics. Now for us to understand heredity, let us first tackle the terms related to heredity. So for our vocabulary check, first we have here the heredity. Heredity refers to specific mechanism by which characteristics or traits are passed from one generation to the next via genes. So again, what is the other term for traits? Okay, characteristics. So for inheritance, inheritance is defined as the passing of genetic information from parent to child. So it is known as inheritance. The information is in chemical codes carried by deoxyribonucleic acid or the DNA in the sex cells or the eggs and the sperm. Now variation. Variation is the differences between individuals within a species. This can be caused by inherited or environmental factors. Variation can be continuous or discontinuous. In biology, any difference between cells, individual organisms, or groups of organisms of any species caused either by genetic differences, genotypic variation, or by the effect of environmental factors on the expression of the genetic potentials, or the phenotypic variation is defined as the variation. Now let's move to genetics, genes, and alleles. So for genetics, it is defined as the science that deals with the study of heredity. And when we say genes, it is a unit of heredity which is transferred from a parent to offspring and is held to determine some characteristics of the offspring. And finally, the alleles. Alleles is the different versions of a gene. Alleles are described as either dominant or recessive depending on their associated traits. Okay, now let's move on to Mendel's laws of heredity. So why we look the way we look? Okay, now let's start with the Mendelian genetics. Mendelian genetics describes inheritance patterns based on complete dominance or recessiveness. So, dalawa lang yung Mendelian genetics, complete dominance or recessiveness. Complete dominance is a type of dominance in which one allele in a single gene pair manifests itself fully in the heterozygote to the complete exclusion of the other, contrasting allele. It was discovered by Gregor Mendel in his monohybrid study with Garden P. For recessiveness, in genetics, the failure of one of a pair of genes present in an individual to express itself in an observable manner because of the greater influence or dominance of its opposite acting partner. Both alleles affect the same inherited characteristic, but the presence of the recessive gene cannot be determined by observation of the organism. So let's now proceed to non-Mendelian genetics. So for Mendelian genetics, we have two, the complete dominance and the recessiveness. But with non-Mendelian genetics, we have five. So not all inheritance is based on the rules of complete dominance. These are the other types of inheritance that Mendel never considered. So first, incomplete inheritance. It is when a dominant allele or form of a gene does not completely mask the effects of recessive allele, and the organism's resulting physical appearances shows a blending of both alleles. So for incomplete inheritance, you can still see the recessive allele. So both the recessive and the dominant allele is present in the physical appearance of the organism. So for codominance, it occurs when two versions or alleles of the same gene are present in a living thing, and both are expressed. Instead of one trait being dominant over the other, both traits appear. So codominance is easy to spot in plants and animals that have more than one pigment color. Multiple alleles. So multiple alleles exist in a population when there are many variations of a gene present. In organisms with two copies of every gene, also known as diploid organisms, each organism has the ability to express two alleles at the same time. So for polygenic traits, these are traits that are controlled by multiple genes instead of just one. The genes that control them may be located near each other or even on separate chromosomes. Now for our last non-Mendelian genetics, we have here the sex link. Genes that are in the sex chromosomes and that are therefore inherited differently between males and females. In mammals, where the female has two X chromosomes and the male has one X and one Y chromosome, recessive genes on the X chromosome are more often expressed in males because their only X chromosome has this gene, while females may carry a defective recessive gene on one X chromosome that is compensated by a healthy dominant gene on the other X chromosome. Common examples of sex-linked genes are those that code for color blindness or those that code for hemophilia or the inability to make blood clots in human. For this slide, let's talk about the dominant and the recessive alleles. So what are the dominant and the recessive alleles? So dominant alleles show their effect even if the individual only has one copy of the allele, also known as being heterozygous. So for example, the allele for brown eyes is dominant, therefore you only need one copy of the brown eye allele to have brown eyes. Although with two copies you will still have brown eyes. If both alleles are dominant, it is called codominance. The resulting characteristic is due to both alleles being expressed equally. An example of this is the blood group AB, which is the result of codominance of the A and B dominant alleles. So for recessive alleles only show their effect if the individual has two copies of the allele, also known as being homozygous. For example, the allele for blue eyes is recessive, therefore to have blue eyes, you need to have two copies of blue eye allele. What is a genetic disorder? So let us now discuss the genetic disorder. So a genetic disorder is a disease that is caused by a change or mutation in an individual's DNA sequence. A genetic disorder is an illness caused by changes in a person's DNA. These mutations can be due to an error in DNA replication or due to environmental factors such as cigarette smoke and exposure to radiation, which cause changes in the DNA sequence. The human genome is a complex set of instructions, like a recipe book directing our growth and development. However, unlike a printed book, the human genome can change. These changes can affect the individual bases, which is the A, C, G, or T, or much larger chunks of DNA or even chromosomes. So in additional, ACG and T is an acronym for the four types of bases found in a DNA molecule. So A stands for adenine, C stands for cytosine, G stands for guanine, and T stands for thymine. Our DNA provides the code for making proteins, the molecules that perform most of the functions in our body. However, when a section of our DNA is changed in some way, the protein codes is also affected and may no longer be able to carry out its normal function. So depending on where these mutations occur, they can have little or no effect, or may profoundly alter the biology of cells in our body, resulting in a so-called genetic disorder. Now let's discuss the genetic disorders that can be grouped into three main categories. So the genetic disorder is categorized into three main groups. First category, we will discuss the single gene disorders. So, single gene disorders are disorders caused by defects in one particular gene, often with simple and predictable inheritance patterns. So, we have three: the dominant diseases, recessive diseases, and the X-linked disorders. So, let us first tackle the dominant diseases. Dominant diseases are single gene disorders that occur when an individual has one altered copy of the relevant gene and one healthy copy. For example, Huntington's disease. So for our next disease, we have here the recessive diseases. Recessive diseases are single gene disorders that only occur when an individual has two altered versions of the relevant gene. For example, cystic fibrosis. And lastly, we have here the X-linked disorders. It is also a single gene disorder that reflects the presence of an altered gene on the X chromosome. X-linked disorders are more common in males because they only have one X chromosome. As a consequence, males only need one copy of the altered gene for symptoms to occur. So for example, the muscular dystrophy. Now let's talk about the second category, the chromosome disorders. So when we say chromosome disorders, it is a disorder resulting from changes in the number or structure of the chromosomes. So for example, the Down syndrome, which results from an extra chromosome 21, the trisomy 21. Okay, trisomy 21 are 3 copies of chromosome 21. So Down syndrome is a chromosome disorder resulting from an extra chromosome 21. So for our third category, we will now discuss the multifactorial disorders or complex diseases. So these are disorders caused by changes in multiple genes, so often in a complex interaction with environmental and lifestyle factors, such as diet or cigarette smoke. So for example, or the most common example for this disorder is the cancer. Now let's discuss more about the genes, the alleles, and the traits. So a gene is a piece of genetic code that determines a trait in a living organism. So for example, the eye color. But eye color can be blue or brown or various other colors. So these variations of the same gene are called alleles. Now let us understand more the genotype and the phenotype. So you will encounter these two later on our discussion with the Punnett square. So again, let's understand the genotype and the phenotype. All our genes, all your genes, together make your genotype. The entire length of DNA that describes how to build you is the genotype. Your actual body and behavior are your phenotype. How you ended up, partly because of genes but also because of diet, possible injury, and other life experiences, so those are the phenotype. Now let's learn about gene inheritance. So in sexually reproducing organisms, including humans, each parent passes on one gene for each trait. The child keeps the genes from both parents. For each trait, the child might have 2 copies of the same allele or 2 different alleles. An organism with 2 copies of the same allele is homozygous for that gene. An organism with 2 different alleles is called heterozygous for that gene. Okay, now let's discuss the dominant and the recessive genes. So let us understand more the dominant and the recessive genes. The simplest genes have 2 alleles, one dominant and one recessive. The dominant variation shows up even if a recessive allele is also present. A biologist would say that the dominant allele is expressed in the phenotype. So again, the phenotype is the appearance of the organism. So an organism with 2 dominant alleles is homozygous dominant. An organism with 2 recessive alleles is homozygous recessive. So 2 alleles of the same gene that can combine to make 3 different colors are called incomplete dominants. So an example of these are cream dilute horses. So where two small letter C, so that 2 small letter C is called the homozygous recessive. And so where 2 small letter C or the homozygous recessive horses are red, and the 1 capital letter C with 1 small letter c, which is called the heterozygous horses, are a shade of gold. And the 2 capital letter C is also called the homozygous homozygous dominant horses are a light shade of cream. So now let's proceed to the Punnett square. So the Punnett square is a square diagram that is used to predict the genotypes of a particular cross or breeding experiment. It is named after Reginald C. Punnett, who devised the approach. The diagram is used by biologists to determine the probability of an offspring having a particular genotype. So we have a diagram or a picture here showing the easy way to learn Punnett squares. So as you can see, the capital letter B is the one that is called the dominant, and the small letter b is called the recessive. So that is the one we call recessive, the small letter b. So when the capital letter B and the small letter b comes together or combine, they are called the heterozygous. But when the two small letter b combine, that's when they are called the homozygous. So why Punnett squares are useful? So let us now discuss the importance of the Punnett square. So the end result of a Punnett square is a probability. A 25% chance at red hair doesn't mean that exactly 25% of the children will have red hair. It's just an estimate. However, even a rough prediction can be informative in some situations. So first, someone running a breeding project, usually developing new plant strains, wants to know which breeding pair gives the best chance at good results. So, or whether a certain breeding pair is worth the effort. And for this other case, someone with a serious genetic disorder or a carrier of an allele for a genetic disorder wants to know the possibility that he'll pass it on to his children. So that's when the Punnett square enters the picture. So that's when it is useful. When you want to know the probability of a certain case, you can use the Punnett square. Okay, so we have here an example. So the key vocabulary here or the terms here is on the other side. So for the big letter T, it is or it means the tall. So for the small letter t, it means the short. So tall, that is the dominant trait, and the short t, it is the short or the small letter t, it is the recessive trait. Now let's proceed to the types of genotype. So the genotype and the phenotype, again you will encounter this later on our next few slides. Okay, let's start with the purebred. So for our purebred homozygous dominant, the genes only have the dominant trait in its code. So for example, the dominant tall or the two capital letter T. So that is called the purebred dominant. So for our purebred homozygous recessive, the genes only have the recessive trait in its code. So for example, the recessive short or the two small letter t. And lastly, we have here the so-called hybrid. So hybrid is also called the heterozygous. So the genes are mixed code for that trait. So as you can see here, both the dominant and the recessive trait are present. So for example, the hybrid tall. So it is still called the hybrid tall. It's still called tall because the dominant T is present. The dominant tall is present. So now let's move on to the Punnett square practice. So let's try to test your knowledge. Now for our first Punnett square practice. So for our first Punnett square problem, we have here a two diagram. So the first Punnett square represents the F1 generation, and then the second Punnett square represents the F2 generation. So the big letter W means the white flowers, and the small letter w means the purple flowers. So we will encounter questions about this on our next slide. So let's for our first question, which trait or characteristics, white flowers or purple flowers, is controlled by a dominant allele, and which is controlled by a recessive allele? So as you can see, I posted the diagram here on the side. So the answer is the answer is the white flowers, or the big letter W is the one that is controlled by the dominant allele, and the purple flowers, or the small letter W, is controlled by the recessive allele. Our number 2 question, in which generation are the parents purebred, and in which generation are they hybrids? So as you can see, we still have here the diagram of the Punnett squares. So kung inyong maobserbahan, we have two generations for each Punnett square, the F1 generation and the F2 generation. So now let us distinguish which generations is purebred and which one is hybrid. So the answer is the answer is the F2 generation shows the purebred and the F1 generation shows the hybrid. So why is that? So as you can observe, F1 generation, the genes here are mixed. So naatay capital letter W and the small letter w, which represents the hybrid or the heterozygous trait. So for the F2 generation, both the dominant trait and the recessive trait are present. So the two big W letter represents the dominant trait, and the two small W letter represents the recessive traits. So both dominant and recessive traits are present in the F2 generation. So it is called the purebred. For our third question, and the last question also, in the F1 generation, what is the genotype offspring of the offspring, and what is their phenotype? So the F1 generation's genotype and phenotype is, so the answer is, the genotype is the hybrid, the capital letter W and the small letter w. So and the phenotype is white flower. So for our next practice, we have here the phenotype and the genotype. So we have three categories for this. So we have the trait or the characteristics, the dominant gene, and the recessive gene. Under trait or characteristics, we have the body shape, body color, eye shape, and nose style. And under dominant gene, we have the Squarepants. So for Squarepants, the capital letter S represents it. And for yellow, we have here the yellow for big letter Y. And for round, we have the big letter R. And for long, we have the big letter L. So under recessive genes, we have the round pants. On the side, we have the small letter S. And for blue, we have the small letter Y, oval, small letter R, and stubby, small letter L. So the questions are the following. Use the information for SpongeBob's traits to write the phenotype for each item. So, ang hinihingi dito yung phenotype o yung appearance. So, for the dominant trait or the double letter L, what is the phenotype or the appearance it represents? We also have here the dominant trait, the double R. Then, we also have here the recessive trait, the two small letter S, and then we have this two small letter Y, another recessive trait. And for our fifth item here, we have the hybrid. And then we also have another hybrid trait, and then another hybrid trait, and lastly we have the recessive trait. So you have to be able to identify its phenotype. What does it represent? So the answer is, okay, here is the answer. So for our dominant trait, first dominant trait, the answer is long. And for our second dominant trait, which is the 2 capital letter R, the phenotype is round. And for our recessive trait, which is the small letter S, 2 small letter S, the phenotype is the round pants. And we also have here for our recessive trait Y, the answer is blue. And for our hybrid, the answer is round. So, anong dili man oval ang answer? Nga naaman diri ang small letter R. Because the dominant gene or the dominant trait here is present, which is the big letter R. And same goes with the hybrid trait here, which is the small letter Y and the capital letter Y and the small letter Y. It represents the dominant gene yellow. The phenotype yellow. And then also for our dominant trait S, the answer is Squarepants. And for our recessive trait L, the answer is stubby. So that's it for our week 3 discussion. For your reviews and activity, you may want to visit our new GENOTYPE e-learning site, which is the https://guidef.sims.school/lcms. If you have any other question, please don't hesitate to ask me. And so yeah, that's it. Thank you for listening.